Alright, so this is the part where we will be discussing the last 10 years questions of the chapter of biomolecules, and we'll be seeing that which sort of questions generally come from this chapter. So while preparing the chapter, it becomes easier for you to concentrate on the important parts. My name is Ojasvi Singh, and this is the third chapter that I'll be covering for the past 10 years question series, and these are some of the exams that I cleared. All right, so let's move directly to the questions. Yeah, so lecithin, a small molecular weight organic compound found in living tissues, is an example of. So I have covered all of these important points while discussing the chapter in great details. So if you have seen that video, you will already be knowing that when we are talking about lecithin, it is a phospholipid. It is present in the plasma membrane of um, your cells. So let's move on. The cofactor for the enzyme carboxypeptidase is, so again, if you have seen the video, you would know that there is a chart. No, actually, there's not the chart in the part where they are discussing about your enzymes. When they are discussing about, apoenzyme plus different cofactors, in that sector, this particular statement is written. So that the cofactor for enzyme carboxypeptidase is zinc. All right. Let's move ahead. Inhibition of succinic dehydrogenase enzyme by malonate is a classic example of competitive inhibition, because one more thing is that this is the only sort of inhibition that is mentioned in your NCERT. There are other inhibition methods of inhibition as well, like non-competitive or mixed inhibition, but they are not discussed in your NCERT. The only type of inhibition that is discussed in NCERT is competitive inhibition. Then we have to match the list. So GLUT4 is, it enables glucose transport into cells. Insulin is an trypsin is an enzyme. Insulin is a hormone, and collagen is your intracellular ground substance. So upon matching, A will go with 4. Only upon matching the first option I could have gotten the answer. Now, before we move ahead, I would like to request you all to, as soon as I'm moving to the next question, first pause the screen and try to solve the question on your own. And if once you solve the question, after that you can resume the video so that you can check whether the answer that you have marked and the answer that I am saying is matching or not. In case you make a mistake, you can correct it, and in case you think that I am making a mistake, you can comment in the comment box, so I can also go back and just reread. I may also make a mistake. I'm a human only. So yeah, let's move again. All right, so I have discussed this question while teaching the chapter itself. So over there, I told you to remember cocaine as co-education. So co-ed, instead of ed, you will write co-a. So cocaine will go with erythroxylum. So A3, A3, we already have our answer. But still, let's go ahead. When I was talking about morphine, I told you to remember it as MOPA. That means morphine is coming from papaver, but again morphine is used in your surgery as a sedative. All right, so morphine, effective sedative. So C will go with 1. Then heroin, I told you to remember heroin as also HEPA. MOPA, HEPA, these both come from papaver. So heroin will go with papaver. B4, yeah, it's matching. And the last one, marijuana, will go with cannabis. Maca. Marijuana, cannabis. Alright, let's move ahead. So over here we have to match with which enzyme is responsible for cleaving of which bond. Right. So nuclease will, first let's start with protease. Protease will break down proteins, and the type of bonds present between amino acids in a protein is your peptide bond. So protease will go with peptide bond. So C1. C1, we have two options. All right. Amylase, for example, your salivary amylase is responsible for breakdown of carbohydrates. Excuse me. Carbohydrates have glycosidic bonds, so amylase will be breaking glycosidic bonds, so D3. So we got our answer, which is option C. Lipase will go with ester bond because lipids have ester bonds, and lipase breaks down the lipids. And nuclease finally will break the phosphodiester bond that is present in your nucleic acids like DNA and RNA. Alright, let's move ahead. Regarding catalytic cycle of an enzyme action, select the sequential steps. So we have to arrange the steps of enzyme catalysis. So the first one would be substrate binding to active site. After the substrate binds to the active site, then so first would be E. So between these two, we have to select. And the last would be that the free enzyme is ready to bind with another substrate. So this would be your last. So B should come at end. So this is your correct option. This is how you can save time in the exam as well. But let's just try to solve it taking our time right now. So E then A, substrate enzyme complex formation makes sense. Then D, chemical bonds of the substrate is broken. After the formation of product, there is release of products, after which free enzyme is ready to bind another substrate. Okay, so now we have to match. Unsaturated fatty acids will have carbon-carbon double bond, so B will go with 1. B will go with 1. We have two options remaining. Then protein has peptide bonds A4. A4. Okay, so this is the correct option. A4, B1. B1. The nucleic acid has phosphodiester bond and polysaccharides have glycosidic bond. Let's move ahead. Inulin is a polymer of fructose. We have already discussed this question multiple times while going through the chapter. So if you have not gone through my lecture on biomolecules yet, then I would encourage you to go through that video because we have discussed all of those important points in details over there. Which of the following is not a secondary metabolite? So I've already told you what are primary metabolites, what are secondary metabolites. In this case, lecithin is a primary metabolite because it is always present in the cell, and the metabolites which are always being synthesized are your primary metabolites. Moving ahead. Okay, so you have to now match this. So if you have gone through my lecture, you would know that ricin is a toxin. So D3. D3, we have two options now. And terpenoids, diterpenes, so A2. We have A2 in both of them. Then we have lectin and alkaloid. So I know that concavalin A is a lectin, so B will go with 4. So this is your correct answer. Alright, this again we have discussed in the chapter in details. And finally alkaloid, so codeine will be your alkaloid. Cellulose does not form blue color with iodine because, so over here, leave cellulose. First, let's start with starch. So starch gives you that blue color with iodine because it has helical structure, and in the helical structure the iodine gets captured in the pockets and forms a blue coloration. Cellulose does not have this helical structure, so it does not contain the complex helices and hence cannot hold iodine molecules. Then given below are two statements. A protein is imagined as a line, the left end represented by first amino acid. So it is saying that the left end starts with C, then amino acid 1, then amino acid 2, then so on, then amino acid N, and then N terminal. And the second statement is saying adult human hemoglobin consists of 4 subunits, 2 alpha and 2 beta. Alright, so if you have gone through that video, you must be knowing that this is not how a protein is represented. In its primary structure, it always starts with the N terminal and then it goes like this. So everything is okay, but the only thing is that it does not start with the C terminus, it starts with the N terminus. Alright, so first statement is wrong However, the second statement is correct because hemoglobin is indeed formed of 4 subunits, 2 of alpha, 2 of beta. So statement 1 is false and statement 2 is true. This would be your answer. Moving ahead, low temperature preserves the enzyme in a temporarily inactive state, whereas high temperature destroys enzymatic activity because proteins are denatured by heat. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme, it is known as competitive inhibitor. Alright, so this is true. So, for example, even in the labs when we are doing experiments, we store all of the enzymes at -20 because when you store it at a lower temperature, it preserves the structure. However, the enzyme is not functional. So the enzyme activity is very low, but the enzyme structure is maintained. Alright, so it is in the temporarily inactive state, but the enzyme is preserved. But the high temperature breaks down the peptide bond, so the enzyme is deactivated completely. So this is true. Destroys them. Okay, and the second statement is also true because when inhibitor closely resembles the substrate, this is the property of your competitive inhibition. So both the statements are true. Yeah, this option. When a carrier protein facilitates the movement of two molecules across the membrane, it is known as symport. Let's move ahead. Okay, so here we have to match. So adenine, as you know, it is a purine, which is a uh, nucleotide. Then anthocyanin, chitin is a polysaccharide. We have studied anthocyanin is a pigment, codeine is a alkaloid. Alright, so match it. And then proteins are also known as primary proteins are also called as polypeptides because they can assume many conformations. They are linear chains. They are polymers of peptide monomers. Successive amino acids. This is the correct option. So they can assume many conformations has no relation to being a polypeptide. Then they are linear chains. It makes sense, but polypeptides being a linear chain does not have any relation. Then they are polymers of peptide monomers. This word is wrong. They are polymers of amino acids. Alright, so this is how you solve these questions, and this is the same thing that N terminal, then amino acid 1, amino acid 2, amino acid 3, amino acid N, and C terminal. Alright, now which of the following pair represents the free living nitrogen fixing aerobic bacteria? So over here you have to focus on two words: nitrogen fixing, aerobic bacteria, and free living. Excuse me. So basically, you have to focus on three words. This is not technically a part of the biomolecules chapter, so it's okay if you don't know this one, but you can look it up. The correct answer would be azotobacter and Mejoringia. Now choose the correct enzymatic reaction. Dipeptidase amino acids. This is correct. Maltose. This is incorrect because maltose breaks down to give you glucose. It's made up of glucose monomers, right? So it's like just glucose and glucose. Two glucose makes maltose. So this fructose over here is making it the incorrect option. And since we have to select the incorrect enzymatic reaction, so this would be our answer. Alright, let's move ahead. If DNA contains sulfur instead of phosphorus and proteins contain phosphorus instead of sulfur. So if you know that there are some amino acids in proteins, for example, your methionine and cysteine, these two amino acids contain sulfur. But DNA does not have sulfur, DNA has phosphorus. Right, in terms of phosphate groups that is attached to the 5' carbon phosphate group. In Hershey and Chase experiment, what they did was they used a bacteriophage and they tried to see that the bacteriophage, what material was the bacteriophage transmitting inside the bacteria? So Hershey and Chase found out that it was the DNA, because they used radioactive phosphorus in that case. So when they used radioactive phosphorus, they found that the bacterial cells had a lot of radioactive phosphorus inside them, which had originally come from the bacteriophage. So the bacteriophage transmitted its radioactive phosphorus containing DNA to the bacteria, and those bacteria then contained that radioactive phosphorus. And after taking out the DNA from the bacteria, Hershey and Chase realized that it's the phosphorus that has been given by the bacteriophage to the bacteria, meaning that it was the genetic material was basically your phosphorus containing material, which was your DNA, right? If it was proteins, then the sulfur would have come down to the bacteria level, but the sulfur remained with the bacteriophage itself. Sulfur did not pass from the bacteriophage to the bacteria. If you don't know Hershey and Chase experiment, you can comment. I will make a separate video on Hershey and Chase experiment, or you can just go and check out. There are already a lot of videos on the experiment. In case it's still not clear, you can reach out to me. So the gist of that experiment was that they had substituted DNA with radioactive phosphorus, and hence they got phosphorus inside the bacteria. So in this question, they are saying that if the DNA contained sulfur instead of phosphorus, and protein contained phosphorus instead of sulfur, what would have been the outcome? So the bacterial cells, since they take up the DNA, and now the DNA has radioactive sulfur, so uh, there would have been radioactive sulfur in the bacterial cells instead of radioactive phosphorus. All right, let's move ahead. So that now becomes a question that is linked to biomolecules, because you should know that in the biomolecule of nucleotides, phosphorus is present. However, in your proteins, sulfur is present because of those two amino acids. All right. So given below are two statements. Amino acids have a property of ionizable nature of NH2 and COOH groups. Hence, they have different structures and different pH. Amino acids can exist as zwitterionic form at acidic and basic pH. So, in this statement, amino acids have a property of ionizable nature, which is true, right? Because amino acids look something like this: NH3 plus, COO minus, H, and R group. So, in this case, NH3 plus is an ion, COO minus is an ion. So amino acids have a property of ionizable nature of NH2 and COH groups. It's true. So this is a true statement, which have because of which they have different structures at different pH, right? Because at an acidic pH, what will happen, that this COO minus would become COOH. So since the minus will be removed, its property will completely change. Now it's no more a neutral compound. Then it will become a positively charged compound, right? Because at a lower pH, I'll just write, at a lower pH, its structure would be something like this: CHNH3 plus and R. So if you look over here, the only charge that this amino acid now has is a plus charge. So because of this charge, the amino acid's property will change, because of which it will have a different structure at different pH. That is the reason why your enzyme activity changes with change in pH, with your optimum activity being at pH equals to pi of the protein. Alright, then amino acids can exist as zwitterionic form at acidic and basic pH. This is false, this is wrong, because zwitterionic form exists only at one pH, which is also known as your PI or isoelectric point. Alright, so if you change it to an acidic or a basic pH, the zwitterionic form will no longer exist. The zwitterionic form of an amino acid is the form where both of these plus charges as well as the minus charge is present. Alright, and the total charge of that particular protein is 0. It's a neutral protein. That is the zwitterionic form. Alright. So over here, Statement 1 is correct, but statement 2 is incorrect. This is the correct option. Let's move forward. So, lecithin is found in the plasma membrane is a glycolipid. It's wrong because it is a phospholipid. We have just studied so many times. Phospholipid. Then, saturated fatty acids possess one or more carbon double bonds. It's wrong. Not saturated but unsaturated fatty acids possess. So, these two statements are wrong. So, let's just cancel out any options which has A or B. This is not our answer. This is not our answer. This is not our answer. So, our answer is A, C, D, and E. Right? Gingerly oil has lower melting point, hence it remains as oil in water. It does not dissolve. Then lipids are generally insoluble but soluble in organic solvents. When fatty acid is esterified with glycerol, monoglycerides are formed. In the enzyme which catalyzes the breakdown of H2O2, the prosthetic group is. So if you have gone through my lecture, you would know that the correct answer to this is heme. Alright, let's move ahead. So, alright, let's start with this. So, glycogen is what? Glycogen is a storage product. Uh, Steroids are hormones. Thrombin is a biocatalyst. So, and globulin is your antibody. So, this was pretty easy, right? Because glucose is stored in animal body in the form of glycogen. Hence, it is a storage product. Then steroids are hormones. I think you must be knowing from animal physiology. Even if you don't, you must be knowing thrombin, right? It plays a role in your uh, clot formation, blood clotting, right? So it's an enzyme which enhances the clotting mechanism. All right, so a dehydration reaction links two glucose molecules to produce maltose. So 2C6H12O6 plus C6H12O6 will give you maltose. So we have to find out the formula for maltose. So whenever two monosaccharides attach, a water is a water comes out because of the formation of glycosidic bond, right? So if H2O is minused from this particular thing, so it will become C12H22O11. So C12H22O11, where is the, yeah, so no, I've marked the wrong answer. Just give me a second. C12H22O11, yeah, this is the correct answer. Moving ahead, we have again a matching. So proteins have peptide bonds. Unsaturated fatty acids have carbon-carbon double bonds. Nucleic acids have phosphodiester bonds. Polysaccharides have glycosidic bonds. We have discussed all of this in great details in the lecture. So if you want a broader explanation, please go and watch the lecture. Which of the following are not secondary metabolites in plants? So again, the difference between a primary metabolite and a secondary metabolite is that the primary metabolite is always being produced, it is always being used, right? So over here in all of these cases, amino acids and glucose is always needed by the plant. So it becomes a primary metabolite. So this is your answer. Alright, because they are asking which are not secondary metabolites, which means which are primary metabolites. Hence, this is the answer. Moving ahead, following are the statements with reference to lipids. So lipids have only single bonds are known as unsaturated fatty acids. This is wrong. Lipids which have only single bonds are saturated actually. Then lecithin is a phospholipid. It's true. Then dihydroxypropane is glycerol. Palmitic acid has, this is false. Palmitic acid has 16 carbon. We have discussed this in details. Okay. So any option which has 1 and 4 are incorrect. So this is incorrect. This is incorrect. Then we have to choose between 3 and 5. Arachidonic acid is 16. This is also wrong. Arachidonic acid actually has 20 carbons. So the correct answer is 2 and 3 only, this one. Identify the substance having glycosidic bond and peptide bond respectively. So the first one has glycosidic bond, which can be your cellulose inulin or chitin, and the second one has a peptide bond. So lecithin is a phospholipid, it won't have a peptide bond, it will have an ester bond. Insulin is a protein, so this is our answer. Cholesterol again is a lipid, so it will have ester bond and not peptide bond. Moving ahead, inhibitor of catalytic activity possess peptide bonds. This question looks incomplete. We have to match, but match it to what? Okay, so this question is incomplete. Anyway, let's try to find out. So inhibitor of catalytic activity, that means it's a competitive inhibitor. So I think it will be malate over here. Then possesses peptide bonds, any protein. So whichever example is a protein will come here. Cell wall material in fungi, it is chitin. Then secondary metabolite, there are many secondary metabolites, so the list goes on: rubber, vinblastin, vincristin, multiple secondary metabolites. Antibodies are also, I mean antibiotics are also secondary metabolites, so it's a long list. Which one of the following is the most abundant protein in animals? Most abundant protein, collagen. Let's move ahead. Identify the wrong statements with reference to the gene I. Allele I does not produce any sugar. Gene I has 3 alleles. When IA and IB are present together, they express same. This is incorrect because IA expresses a different type of sugar and IB expresses a different type of sugar. They don't express the same type of sugar. Identify the basic amino acid from the following. Okay, so while discussing the chapter, I have explained the properties of different amino acids. Some amino acids are categorized as basic amino acids, whereas some amino acids are categorized as acidic amino acids. Over here, it is asking you to identify the basic amino acids, so it will be lysine. There are two amino acids which have a basic nature, which is your lysine and arginine. All right, let's move ahead. Okay, so the question is saying that coenzyme of a metal ion that is tightly bound to the enzyme protein is known as prosthetic group. So over, uh, like while you were reading the NCRT, we came across this line, and it's the exact line that is present in the NCRT, that when metal ion is tightly bound to the enzyme, it is known as prosthetic group. So it's a true statement. The second statement is a complete catalytic active enzyme with its bound prosthetic group is known as apoenzyme. This is wrong. Basically, a complete catalytic active enzyme is known as holoenzyme and not apoenzyme. In fact, apoenzyme is incomplete. Apoenzyme with either a prosthetic group or a cofactor or um, a metal ion is basically your holoenzyme or catalytically active enzyme. So statement A is true, but B is false. Concavalin A is what? It is a lectin. Concavalin A is a lectin. All right, let's move ahead. So which of the following statements is correct with reference to enzymes? So holoenzyme is apoenzyme plus coenzyme, as we just studied in the last slide. Which of the following are not polymeric? Lipids. Lipids are not polymers. Protein is a polymer of amino acids. Polysaccharide is a polymer of monosaccharides. And nucleic acid is a polymer of, like DNA, etc., is a polymer of nucleotides and nitrogenous bases. Two functional groups characteristics of sugars are sugar has a carbonyl and a hydroxyl group. All right, let's move ahead. So a non-proteinaceous enzyme, an enzyme which does not have a protein, is your ribozyme. It has RNA instead of protein. Which of the following is least likely to be involved in establishing the 3D folding? So when we are talking about the 3D folding, it has sulfur-sulfur bond, it has hydrogen bond. However, ester bonds are not there in the proteins. Ester bonds are in your lipids and not proteins. All right. Which of the following describes the given graph? So over here, as you can see, the substrate has higher energy compared to the product. That means the energy has been dissipated outside, which makes it a exothermic reaction. So it is a exothermic reaction in presence of enzyme and B in absence of enzyme. So this is the correct option. A in the presence of enzyme and B in the absence of enzyme, because in your B, the activation energy is high compared to A. So when the activation energy is high, it means that the enzyme is not there. If the activation energy is significantly reduced, it means that there is an enzyme present. A typical fat molecule is made up of uh, one glycerol and three fatty acid molecules. Which of the following biomolecule does have a phosphodiester? So which has a phosphodiester bond? So it would be nucleic acids. Then which of the following statements is wrong? Uracil is a pyrimidine. Glycine is a sulfur containing. This is wrong. Glycine structure is this: COO minus NH3 plus H and H. So it does not have any sulfur. In fact, glycine is the simplest amino acid because it has hydrogen present in place of its R group, right? The only two amino acids which contains sulfur are your cysteine and methionine. To remember this, I would advise you to go and look at the structures of all the 20 amino acids. Once you look at the detailed structure, you will realize which amino acids have sulfur, which amino acids have hydroxyl group, which amino acids are aromatic in nature. Okay, so there are three amino acids which are aromatic. One is your tryptophan, one is phenyl alanine, and the last one is your tyrosine. These three are known as aromatic amino acids. Alright, so let's move ahead. Uh, Select the option which is not correct with respect to enzyme action. Substrate binds with enzyme at its active site. Addition of lot of succinate does not reverse the inhibition. So this is wrong, because if you add a lot of succinate, it will outperform the malate. It will go and bind to the active site faster than the malate because it is more in number. Okay, so which one of the following is incorrect? The competitive inhibitor does not affect the rate of break. The presence of competitive inhibitor decreases. So this is wrong. Instead of decrease, it should be increase. So in the presence of competitive inhibitor, the Km increases. The reason for this is discussed in details in the lecture of biomolecules. So if you want to know and understand it better, you have to go and watch my lecture on the chapter of biomolecules. The chitinous exoskeleton of arthropods is formed by the polymerization of N-acetylglucosamine. So this was it from my side. These are all the questions that I had prepared for you for the last 10 years. So if you have any confusion, please feel free to ask in the comment section. You can also reach me at my email ID, which is given. One more thing is that please like, share and subscribe, because I will be only posting this content as long as I'm motivated, because technically I'm not selling any course or anything. So the only key driver which is pushing me to make and upload these videos is motivation from you all. So if I do not see enough views and comments, I will feel like my videos are not as useful, and if they are not useful, there's no point in making those videos, right? So yeah, so just keep me motivated to make more videos for you all. Please like, share and subscribe, and all the best. Study hard. I hope your exam goes well. All right, chalo, bye.